There you go. Thank you so much for clicking this video to watch my presentation. It really means a lot. My name is TJ Rogers. I am a fourth-year PhD student in the Lloyd Lab at the University of Tennessee. And today I'm going to talk about the subsurface landscape of a chemoautotrophic-based ecosystem in the Costa Rica Subduction Zone. Currently, we know there are 1.85 billion gigatons of carbon on Earth, and 99.8% of this exists below ground. Subduction zones provide a conduit that connects this carbon from the surface to the surface world. To give you a lay of the land, a subduction zone consists of an oceanic plate that subducts below a continental shelf. As this plate subducts, it releases fluids rich in iron, sulfur, hydrogen, and CO2. Now, as this volatile-rich fluid is released from the subducting slab, it percolates up through cracks and fissures in the overriding plate and can be expressed on the surface in the form of hydrothermal systems such as hot springs and mud pots. Now, within 122-degree isotherm of the subsurface of the subduction zone, we expect to find microbial populations. This 122-degree isotherm is based on the currently known upper limit of cell proliferation. As this volatile-rich fluid percolates up through the overriding plate, microbial populations living within the subsurface can use these volatiles to power the biosynthesis of CO2 into biomass. However, very little is known about how microbes interact with the carbon and other volatiles as they move between the surface and subsurface world. So we want to know if we can get an idea of how these microbes interact with these tectonic processes. So the goals of this project are threefold. First, we wanted to characterize the subsurface metabolic and taxonomic landscape across the Costa Rica subduction zone. Then we wanted to identify the major carbon fixation pathways in the deep subsurface. And then finally, we wanted to determine what energy pathways power chemolithoautotrophy within the subsurface system. To get at this, our lab, along with collaborators, took a trip down to Costa Rica and sampled from multiple hot springs across this Costa Rica subduction zone. We collected sediment and fluid samples. From these samples, we extracted all the DNA and we sequenced them and then constructed metagenomic assemblies. We then took those metagenomic assemblies and binned them, creating metagenomic assembled genomes, or MAGs for short. We were able to taxonomically identify, functionally annotate, and get the relative abundance of each of these MAGs. And after this process, this is what we were left with: 404 medium to high quality MAGs. Now, 63% of the total bin reads were found in what we're calling the province-specific clusters. This includes the forearc and arc cluster and the outer forearc cluster, while another 37% of the total bin reads were found in what we're calling the prevalent cluster. They're called prevalent because these MAGs didn't have any distinguishable distribution patterns across the subduction zone. Now, after looking at taxonomic identification, looking at metabolic predictions, and looking at where representatives of these MAGs are found, we found that this prevalent cluster represents a surface to shallow subsurface community. And for the purposes of this talk, we're only going to focus on the province-specific clusters. The way we structured this heat map was we first constructed a Spearman rank correlation-based matrix and used that to construct a dendrogram examining the relationships between sites based on their MAG abundance makeup. We then constructed another Spearman rank correlation-based matrix of the bin abundance across the sites. So just like the sites, two bins that are close to each other in this dendrogram, their abundance distributions are more alike than two bins that are separate from each other. Now, with these two dendrograms used together, we can construct a heat map of the abundance across the sites, and we can start to see distinct patterns form. For example, if you look up at the top of this heat map, we can see all these green sites, which represent outer forearc samples, all cluster together, whereas our forearc and arc sites cluster separate from those. We can see that there are MAGs that are only found in the arc and forearc, and then MAGs that are only specifically found in the outer forearc. We can even look at the metabolic predictions and start seeing distinct patterns emerge. For example, in the outer forearc, we have an abundance of genes associated with hydrogen utilization, whereas in the arc and forearc, we have more aerobic associated genes. These are going to be cytochromes. The top 10 classes that are associated with these clusters, these province-specific clusters, we find that representative members are predominantly recovered from geothermal, aquifer, and deep subsurface systems. Coming back to the heat map, we wanted to get an idea of what the different geochemical parameters were that may be driving these distribution patterns in these province-specific clusters. For this, we turned to a prior research project that was carried out by a previous grad student, Kate Fullerton, in which they looked at the distribution of 16S rRNA amplicon sequences across the same subduction zone. They found that the microbial communities were closely associated with temperature, pH, carbon isotopes, and fluid and sediment associated metals. We took these same variables and we used them to construct a constrained ordination analysis. Specifically, we conducted a transformation-based canonical correspondence analysis, or TBCCA. On this plot, diamond shapes represent sample locations, and they're color-coded based on the province. Orange represents the arc, blue represents the forearc, and green represents the outer forearc. Circles represent individual mags, and they are color-coded based on if they contain a carbon fixation pathway and the type of carbon fixation pathway they contain. Blues for Calvin-Benson, orange is for RTCA, and purple is for Woodland-Dahl. The size of the circle corresponds to the scaled average abundance of that mag across the sites. The TBCCA revealed that temperature, dissolved inorganic carbon, aqueous iron and nickel, phosphate, and sediment-associated aluminum explain 36% of the variation between these sites. Now you'll notice that the outer forearc sites cluster to the left of this plot. The previous study revealed that the outer forearc fluids are deep fluids that are partially equilibrated with feldspar, a mineral that can contain aluminum phosphate. These fluids also contain the lowest amount of aqueous phosphate. Under these conditions, microbial communities can mobilize phosphorus from feldspar. In this case, the correlation with aluminum might be an indication of microbial-induced mineral weatherization of feldspar to make phosphorus more bioavailable. You'll also notice that on the right side of the graph, the forearc and arc sites cluster together. Additionally, the Woodland-Dahl mags that are associated with the arc and forearc are closely associated with nickel and iron. Both of these metals are key metals for the Woodland-Dahl's metalloenzymes. Next, we wanted to see if there were differences in the distribution patterns of metabolic processes in each of these clusters. The outer forearc is characterized by serpentinization, and serpentinization produces hydrogen and sometimes hydrogen sulfide, and this increases the pH of the system. And we find that the metabolic predictions reflect the geochemical parameters of the outer forearc. For example, the dominant electron donor was predicted to be hydrogen, and the dominant metabolic pathway turned out to be sulfate reduction. When we look at the forearc and arc, we find that the abundance of sulfur oxidizing and nitrogen cycling genes suggests that sulfur-based denitrification is the dominant metabolism within these provinces. You'll also notice that there is an increase in aerobic respiration when compared to the outer forearc. And this may be due to the fact that as we increase distance from the trench and approach the volcanoes, the 120-degree isotherm becomes shallower, increasing the likelihood of atmospheric influence on the microbial communities within these provinces. When we look at carbon fixation pathways within these province-specific mags, we find that 29% of them contain carbon fixation pathways. 8 contain the Calvin-Benson cycle, 46 contain the Woodland-Dahl, and 7 contain the reductive citric acid cycle. However, we see that Woodland-Dahl dominates in all 3 provinces, and RTCA only comes in a little bit in the outer forearc. However, this turns out to be due to only one mag, sulfur-hydrogen-nebium, and that is very abundant in only one of the sites. So for the purpose of this talk, we're only going to focus on the Woodland-Dahl in all 3 provinces. When we look at the outer forearc, we find that the top 3 classes of Woodland-Dahl containing mags are all acetogenic. We can also see an abundance of genes associated with hydrogen, hydrogen sulfide, and sulfite for electron donors, and sulfate as an electron acceptor, suggesting that the most abundant autotrophic metabolism here is chemoautotrophic sulfate reduction. In the forearc, sulfite oxidation has the highest abundance of genes, followed by hydrogen and hydrogen sulfide oxidation. And then when we look at electron acceptor predictions, we see that nitrite reduction has the highest abundance of genes, followed by nitrous oxide reduction and sulfate reduction. And this suggests that sulfur-based denitrification followed by sulfate reduction are the highest abundant metabolisms in the forearc. And we see that the same metabolisms are also abundant in the arc. So to bring this all together, we found that representatives of province-specific MAGs are subsurface associated taxa. Subsurface provinces are metabolically diverse and reflect local geochemical parameters. And acetogenic carbon fixation via the woodland dominates across the subsurface landscape. In the outer forearc, we find that the main metabolism is anaerobic chemolytic autotrophic sulfate reduction, whereas in the forearc and arc, we have sulfur-based chemoautotrophic denitrification. And with that, I would like to say thank you to everyone that's helped me with this project, and thank you again for your time. It's greatly appreciated.